All right, so this is going to be an introduction to animal diversity. Chapter 33 is going to focus on invertebrates. Chapter 34 is going to focus on vertebrates. This is going to look more at the characteristics that we see among our animals, what they have in common, and how they are able to differentiate. So animals are multicellular, they're heterotrophic, they're eukaryotic, their tissues develop from their embryonic layers. We have identified 1.3 million living species. And there are exceptions for every criteria to be able to distinguish animals from other living things. But when we look at them as a whole, we can identify certain characteristics that can be described that apply to the group. Okay. One is they're nutritional, that they're heterotrophs, that they take in food to get their nutrition. We talked about their multicellular and eukaryotic. They do not have cell walls. They are held together by proteins such as collagen. We have nervous tissue, we have muscular tissue, and the tissues themselves have some sort of common structure or function making them up. They reproduce for the most part sexually, with the diploid stage being the prominent stage throughout your life cycle. Once an egg has been fertilized, the zygote will go through several stages of cell division known as cleavage, and that's how we get the multicellular blastula. The blastula then undergoes gastrulation, which has the different layers of the embryonic tissues, the ectoderm, the endotherm. We also see how the blastopore gets formed and the archenteron. A lot of animals have at least one larval stage. That larva is immature sexually and is definitely morphologically distinct from the adult. It eventually will go through a metamorphosis and form a juvenile form. And the juvenile form is more like the adult, morphologically speaking, but is not yet able to reproduce. Animals are the only ones that will have Hox genes that help to regulate their body form. They don't all have them, but many of them do. And although these are highly conserved, they can result in a wide range of diversity in terms of the morphology of our animals. So this is going back a little bit into our previous unit on evolution. We've got a wonderful diversity of living animal species, and we have an even greater diversity of those that are extinct. The common ancestor is thought to have lived between 675 and 800 million years ago. It may have resembled what we see now as a choanoflagellate. The protists that are most like our animal relatives. So there is kind of a breakdown. Remember, these are part of the unicont clade. Okay. So back in the Neo-Proterozoic era, there were members in the fossil record of the animals that date back to this, which would be 565 to 550 million years ago. In the Paleozoic Era, 542 to 251 million years ago, the Cambrian explosion took place. That's when we started to see lots and lots of groups of living organisms. Why we saw the previous species diminish might have had to do with predator-prey relationships or the presence of increasing amounts of oxygen and then the good old Hox genes. The diversity increased throughout this era. There are mass extinctions that cause there to be significant changes in that diversity. And then they started to really impact on land about 460 million years ago. And they truly made the transition around 360 million years ago. In the Mesozoic Era, 251 to 65.5 million years ago, we had the coral reefs start to form. We had the plesiosaurs ancestors. They were reptiles that went back into the water. And then the dinosaurs were the primary terrestrial vertebrate. We had our first mammals, and then we had the diversity with both the plants that flowered as well as insects. In the Cenozoic Era, the one that we're a part of right now, 65.5 million years ago to now, We had some mass extinctions of both land and aquatic animals, the non flying dinosaurs, marine reptiles. Mammals got larger. They took advantage of ecological niches that no longer had species, and the global climate was able to cool down to increase, that led to increased survival for mammals. Um, we can look at animals and be able to describe them based on how their body is organized, their body plans. This is looking at the morphological features as well as their developmental features. Um, One example of a conserved developmental characteristic would be gastrulation, and we see that among a range of animal groups. So we can categorize animals based on their symmetry or not having symmetry. Some of them will have a radial symmetry. Um, they don't have, so they're going to, you know, be more like what we would see in a circle. They are sessile plantonic. Then we have the two-sided symmetry, which would be us, the bilateral symmetry. We have a dorsal side and a ventral side, a right and a left side, anterior, posterior, cephalization, development of a head. We are more bilateral animals are more likely to be active and to have a central nervous system. So there's an example of radial. So that would be more like a cup or planting, whereas bilateral would be more like a shovel. Um, your body plans also can help to determine the organization of your animal tissues or vice versa. Specialized cells that get isolated from one another by membranous layers. We have three germ layers that help to give rise to the tissues and your organs. We have the ectoderm, which is the one that covers your embryo. The endoderm is the innermost germ layer and lies the digestive tube called the archenteron. Sponges actually, although they are considered an animal, don't have true tissues. Um, Diploblastic animals will have the ectoderm and the endoderm, so those would be the cnidarians and the comb jellies. Triploblastic animals will have a mesoderm involved, and those are all of your bilateral symmetry, animals that have a bilateral symmetry, flatworms, arthropods, etc. The triploblastic animals will often possess a body cavity, which is called a coelom and is derived from mesoderm. Coelomates are animals that have the coelom. The pseudocoelom is a body cavity that's derived from both the mesoderm and the endoderm. Triploblastic animals that have the pseudocoelom are called pseudocoelomates, and triploblastic animals that don't have the body cavity are called acoelomates. Coelomates and pseudocoelomates belong to the same kind of group or grade of species. They share similar biological features. They don't necessarily have to be a clade, so they don't have to have the ancestor and all the descendants in them. So these are examples of this of what we're talking about with the coelom, where they're coming from, and the difference between a coelomate, a pseudocoelomate, and an acoelomate. Ugh, sorry, I had to look up that one. Protostome and Deuterostome development. This is another way we can characterize animals based on these two features. When you are going through protostome development, your cleavage is spiral and determinate. When you're going through deuterostome development, your cleavage is radial and indeterminate. So when you go through indeterminate cleavage, every cell at the beginning can still have the possibility of developing into its own embryo. And that is why we can have identical twins in some animal species. And that is part of the reason why embryonic stem cells are often looked at as something to study to be able to examine how things are being manipulated. Um, Cilium formation and protostome development, when we split in solid masses of the mesoderm, we're able to form that. And the deuterostome, it doesn't just split off, it buds off from the wall of the archenteron. And the blastopore that forms that connects the archenteron to the gastrula's exterior will become the mouth in a protostome, and a deuterostome, it becomes the anus. So there's some examples of the blastopore. You can see that in terms of protostome development versus deuterostome development, same thing with the cilium and with cleavage. So we, as zoologists right now, recognize about 3 dozen different animal phyla. They're not just looking at the morphological data, they're looking at molecular and fossil data. And there are different ways these relationships are explained based on these characteristics. And so we're going to look at two different examples, one that's based on molecular data and one that's based on morphological and developmental. So this is one of these. I believe this one is the morphological. And you can see this one's a little bit different, and this one is based on the molecular. So regardless of which way you're looking at it, things that animals have in common, they have a common ancestor, that sponges are basal, that they are outside because they don't have the true tissues. The eumetozoas are a clade of animals that have true tissues, and most of the animal phyla belong to the Bilateria clade, and the chordates and some other phyla will belong to the Deuterostomia clade. So if we look at the morphology-based tree, the bilaterians are divided into the deuterosomes and the protozomes, and that if we look at it on a molecular level, we see that we have the deuterosomes, the ecdysosomes, and the lophotrosomes. And so, how do we distinguish between those? So, the ecdysoans shed their exoskeletons. The lophotrozoans have a lophophore as a feeding structure. And then we have some that use a trochophore larva stage to go through when they're going through the process of development. And you can see that up there with the picture that I put on the slide. So there's an example of a lophophore feeding structure. Pretty wild. With all that in mind, again, just like we've seen with the other organisms we've looked at, we're going to need to continue to study to be able to better ascertain where everything fits in terms of evolutionary history, and modifications might need to be made.